Now let's move into the analysis of experimental data. First, we will see what will be the expected results for isopycnic separation for the three models that were stated earlier. The extreme left centrifuge tube is actually contains the parental DNA. It's basically have a parental DNA. Consider this pink color as a DNA that contains N15. So you can see a single parental duplex that is in the stock culture, and they all incorporated N15 into their nitrogenous bases. So the duplex that you get in the parental DNA is simply called a heavy DNA, simply because all the bases in this duplex will incorporate N15. So you start from the parental culture. In the parental culture, you have only a single duplex, and in that single duplex, in both strands, all the bases will incorporate N15. So naturally, the density of this DNA will be higher. So you are going to classify it as a heavy DNA. If you do isopycnic separation in the cesium chloride density gradient column, you would expect the DNA to settle or sediment onto the bottom of the tube. Why? Because the DNA is actually much denser. So I'll simply represent a heavy band at the bottom of the tube as in this way. So say this is our, simply imagine that this is our, so I'm simply saying that this is our heavy band. This is our heavy band that corresponds to the heavy DNA. So I think you have understood why you got a single band at the bottom of the tube, because the DNA was denser. So the top of the tube was actually lighter. So the DNA added onto the top of the tube will gradually settle until they are going to sediment onto a layer where their density is equal to the density of the column. Now let's first analyze if the mode of DNA replication was conservative. What will be the expected banding pattern in several generations? So we already know that only the parental generation was grown in the N15 medium, then all the other generations were grown in N14. So here represented are the samples collected from first generation, meaning the sample you have extracted from after 20 minutes. The second generation, meaning 40 minutes, third generation 60, and fourth 80 minutes. So these are the different tubes that corresponds to the DNA extracted at different times during our experiment. Now, what will be the number of dotted duplexes formed after each round of replication? So you can use this formula. The number of duplex DNA produced after replication is simply equal to 2 raised to n, where n stands for the number of generations. So if you look at the first generation, the generation is first one. So the total number of DNA duplex that is formed will be 2 raised to 1, which is 2 duplexes here. So you know that in the first generation replication is actually happening, the E. coli cells are in a medium that contains N14. So all the newly formed DNA will incorporate the lighter isotope. According to the conservative mode of replication, there must be 100% conservation in the inheritance of these nitrogens. Accordingly, you find that there are two dotted duplexes. One of the dotted duplex still contains only N15, and the other dotted duplex contains N14 only. Imagine that the blue color represents N14. So you can actually see that there is 100% conservation. Only one of the dotted duplex contains only N15, whereas the other duplex contains only N14. Since both the strands of this daughter duplex contained N15, still their density is heavy, so you represent it as a heavy DNA. Whereas both strands of this daughter duplex inherit N14, the nascent DNA, so they are basically lighter than the parental duplex. So you should actually have two types of duplexes with two different density. One is heavy as the parental and one is lighter compared to the parental. So how many bands do you expect for the first generation? Is you expect two bands. One band is at the bottom of the tube that corresponds to the heavy DNA, and you expect another band at the top of the tube that corresponds to the light DNA. Their thickness will be the same. Since the concentration of both the duplexes are the same. So you will have a heavy band that corresponds to the heavy duplex and a light band that corresponds to the light DNA. Coming to the second generation, what you find is that it is these two daughter duplex that replicate into the second generation. So what you find is that when this heavy DNA replicate, you will have two daughter duplexes, and one will be the heavy one and the other will be the light, because the new nascent DNA always incorporates the lighter isotope. Whereas the daughter duplex in the first generation, which already has N14, they actually give rise to two daughter duplexes which already have N14. So this will be light and this also will be light. So how many bands do you get? Like the first generation, we get only two types of bands. One corresponds to the heavy band at the bottom, with the same thickness as the first generation, because the concentration is the same. Whereas when you go to the light band, what you find is that the thickness of the light band will actually increase, and the thickness of the light band will increase because the number of lighter DNA duplex has increased. So what is the ratio? What is the expected ratio you find here is that you will get one heavy and three light bands, three light duplexes are obtained. So the heavy to light ratio for the second generation will be 1:3. Coming to the third generation, how many daughter duplex do you get? The third generation means it is 2:3. That means you should actually get 8 duplexes. And who are those duplexes that contribute to this? It is the 4 daughter duplexes in the second generation that replicate to form the 8 duplexes in the third generation. And we already know that from this heavy duplex, you are going to get one heavy and one light daughter duplexes. Whereas from all these three light duplexes, you are going to get 6 light duplexes. So totally what you are going to get will be 1 heavy is to 7 light. You can actually see when you compare to the second generation, you can find that the concentration or the proportion of the heavy band still remains the same, but the concentration of the light duplex has increased 7 times. So the banding pattern still remains the same. In the third generation, you are going to find a heavy band with the same thickness as the second and the first generation. Whereas when you go to the lighter band, it has become more thickened because the concentration has increased 7 times compared to the heavy DNA. So this will be the banding pattern you observe. Now coming to the fourth generation, you can use the same logic. You can see in the fourth generation there will be 2:4 duplexes, which is actually 16 duplexes. And you find that the concentration of the heavy band is still the same. So you are going to find one heavy and the rest 15 duplexes will be light. So you can actually see from the first generation we have started from 1:1. We have started from 1:1 ratio, then it changed to 1:3, then it changed to 1:7, and then finally changes to 1:15. So you can find in the fourth generation also the thickness of the heavy band is going to remain the same, whereas the thickness of the light band has still increased, because now there are 15 times more lighter DNA, 15 times more lighter duplexes than the heavy band. So you can actually conclude that if the replication is actually conservative, apart from the parental generations, all the generations will contain two bands. One will be the heavy band and other will be the light band. As the generation increases, the concentration of the heavy band will remain the same, whereas the thickness of the light band keeps on increasing. Now let's check semi-conservative mode of replication. We have already said that in semi-conservative mode of replications, the parental duplex separate act as a template for the synthesis of the nascent DNA, so that there is 50% inheritance of the parental DNA between the daughter duplexes, as we have already explained. So in the first generation, what you get here will be two daughter duplex, and each daughter duplex will contain the parental DNA, which is actually N15, as well as the nascent DNA, which is N14. So when you look at the density of this DNA, it is going to be lighter than the parental strand, which is a heavy one, whereas it is going to be denser than the light DNA, which contains both the strands with N14. So you will have this DNA with an intermediate density. You will call this as a hybrid DNA. So in the first generation, what you find is that you are going to get two daughter duplexes with the intermediate density called as the hybrid DNA. So where do you expect in the isopycnic separation, where do you expect the band? We have already seen in the conservative mode that one will be at the bottom, that corresponds to the heavy band, and one will be at the top, that corresponds to the lighter one. But here you are going to get a hybrid band. You are going to get a hybrid band. I am simply writing it as a Y band, which is the hybrid band. This will be your expected observation for the first. Now coming to the second generation, you can find that there will be 2:2, there will be 4 daughter duplexes will form. So from this first generation, how many daughter duplexes will form? If you see, you can find that if you consider this daughter duplex alone, its heavy strand can actually give rise to a hybrid one, because the opposite coming complementary strand will be of the lighter N14, whereas the light strand of this duplex will incorporate another light strand during the next round of replication. So this single daughter duplex will give rise to another hybrid DNA and a light DNA in the second generation. Similarly, this hybrid DNA will give rise to two such daughter duplexes. One is a hybrid one and other is a light DNA. So what is the expect? So you are going to find two types of DNA here. One is the hybrid density DNA and other is the light DNA. What is the proportion? You can see it is 50-50. So you can see you are going to find two hybrid bands and two light bands. So you find the ratio as 1:1. Right, two hybrid bands and two light bands, which is in the proportion as 1:1. So when you move on to the second generation, what you find will be you are going to find a light band. You are going to find a light band. A new band will appear in the second generation, and similarly you are going to find a hybrid band with the same thickness as the first generation, because you can see in the first generation you had two hybrid, in the second generation also you have two hybrid. So you will have a hybrid band as well as a light band with the same, both will have the same thickness. Now coming to the third generation, you have 2 raised to 3 8 daughter duplexes will be obtained from the daughter duplexes of the second generation. You can find that it is now easy to calculate from the hybrid, from the 2 hybrid duplexes in the second generation, how many daughter duplexes can you get? 4. And what are the 4 daughter duplexes? We have already seen from the first to second generations that 2 hybrid DNA can result in 2 hybrid and 2 light. So you can actually find that definitely you are going to get 2 hybrid and 2 light from the hybrid duplex, as well as from the lighter duplexes you are going to get 4 light DNA. So totally 8 duplex. Out of that 6 will be light duplex and 2 will be the hybrid one. So so you have 2 hybrid and 8 light, sorry, not 8, it will be 6 light DNA duplexes. So from the ratio in the second generation, which was 1:1, now the ratio is changed to 1:3. So you find that the banding pattern is still the same. You are going to find 2 bands. One is the hybrid band and other is the light band, but the difference is the thickness of the light band is now increased because there are 3 times more lighter DNA than the hybrid one. Now coming to the fourth generation, similarly you are going to get 16 duplexes here, but you can simply calculate, you can find out that there will be no differences in the number of hybrid duplexes formed. Still there will be 2 hybrid duplexes formed, and the rest 14 will be light DNA duplexes. And so you can see that now the proportion is still more separate. You can see now there is 1 hybrid duplex is to 7 lighter DNA. So still now you have 2 bands. One is the hybrid band with the same thickness as the first, second and third, but now the thickness of the light band has now increased because there are 7 times more lighter DNA than the hybrid one. So this is the banding pattern that you are going to observe in the semi-conservative. So what's the characteristic? The characteristic is that in the first generation you are going to find, in the first generation you will find that the heavy band that was observed in the parental DNA is disappearing and a new band is going to appear, which is a hybrid density band. And the second generation, a second band is going to appear, that will be the light band. Thereafter the banding pattern is the same, but the thickness of the light band keeps on increasing from one generation to the next generation. Finally, coming to the dispersive mode of replication, we have said that in dispersive mode of replication there are random double-stranded nicks in the DNA that allow their rotation and resealing, so that every strand of the DNA will contain both the parental as well as the nascent DNA. There is random inheritance of the parental DNA. That is the dispersive model. So what you find here will be from the parental DNA, after the first generation you are going to find 2 daughter duplexes, and when you look at the daughter duplex, what you can find is that each strand contains both the pink as well as the blue, indicating that each strand contains N15 as well as N14. So what will be the density of this daughter duplexes will be? They will be lighter than the parental DNA because they contains more N14 than the parental, but they are more denser than the light DNA because they contain some of the parental N15 also. So like a semi-conservative mode of replication, you are going to find a new band appearing in the first generation, and that will be a hybrid band. Now coming to the second generation, you find that there will be 4 daughter duplexes formed. If the mode of replication is dispersive, still there will be random inheritance of the parental DNA. But because 2 generations has passed by, and for the 2 generations this E. coli is in a medium that contains N14, what happens is that when you look at each strand, there will be more N14 bases incorporated relative to the first generation. Why? For the first generation, the bacteria has remained in that particular media for say 20 minutes only. But when you come to the second generation, they have been there for more than 40 minutes. So meaning that more N14 will be incorporated into these DNA. So what you find here will be that 4 daughter duplexes will still contain the N15 from the parent as well as the N14 from the nascent, but the amount of nascent DNA definitely would have increased for the second generation. So what you are going to get will be still a hybrid band, but the density of the hybrid band is not the same as the first generation because they have incorporated more N14. So you can find that. So this should be the actual position of the hybrid band if the density was the same as the first, but you can see that now the hybrid band is still shifted up, meaning it has become more lighter. This is your observation for the second. Now coming to the third generation, you find that there will be 8 daughter duplexes, and all of these 8 daughter duplexes will be hybrid. Why? Because they still retain the N15 randomly among them from the parent. But what happens is that all these 8 daughter duplexes will now contain more N14 incorporated than the second generation, but their density is still a hybrid one. So you find still a hybrid band whose thickness has now increased, because in the second generation you had only 4 daughter duplexes. Now you have 8 daughter duplexes, so definitely their density, their thickness have increased, but their density has slightly decreased. Why? Because from the second to third generation there were more N14 incorporated. So you can actually find that the band has now become more lighter, and the band has become now more lighter than the second generation. Now coming to the fourth generation, you will have 16 duplexes, and if you follow the similar logic, you find that all the daughter duplexes will be still hybrid, but they have more N14 now than the third generation. So you find that the band thickness has now increased very much, because there are now 16 daughter duplexes in this sample, but it has become more lighter due to the incorporation of more N14, but still the thickness, still the density of the band is actually hybrid. They are more more denser than the typical light band, and they are lighter than the typical heavy band. So when you look at the dispersive mode of replication, you find that in all the generations, including the parental generations, you will find only one band. In the parental generation, that one band is the heavy band, whereas when you come to the daughter generations, first, second and third and fourth, there will be only single band that corresponds to the hybrid band. But the peculiarity is that the hybrid band is found to migrate from the bottom of the tube to the top as generations pass by. Why? Because as generations pass by, more N14 is getting incorporated, so the thickness of the band is increasing, as well as the density is becoming more lighter. So this shows the overall data that is expected for each specific model for replication. So you can see that each banding pattern is characteristic for that particular model. But you can see a special characteristic. When you look at the first generation, if you identify the semi-conservative as well as dispersive model for replication, you can see their banding pattern is the same. That is, for the dispersive as well as the semi-conservative, you get a single band that corresponds to a hybrid density. Whereas for conservative mode of replication, you should get a heavy band as well as a light band. So imagine if the Meselson-Stahl experiment were conducted only for a single generation. If the experiment was conducted for only a single generation, definitely you can discard conservative mode of replication. Why? But you cannot distinguish a semi-conservative from dispersive, because in the first round of replication, because we already know that now the replication is semi-conservative. But if the experiment was conducted only for the first generation, definitely you cannot distinguish a semi-conservative from the dispersive, because their banding pattern is the same. So we can state that in the first generation, the model you can discard is the conservative mode of replication, because you are never going to get two bands for the first generation in the actual experiment, because all DNA replications are semi-conservative. Whereas when you go to the second generations, you will be able to distinguish between a semi-conservative as well as a dispersive model, because in semi-conservative you will get two bands. One is the hybrid as well as the light, and we know for the dispersive, regardless of the generations, you will get a single band only on. So to confirm that DNA replication is semi-conservative, Meselson-Stahl experiment should be conducted at least for 2 generations. In the first generation, you can discard the conservative. In the second generation, you can discard the dispersive model for replication.